Okay, so the reason why we learn a lot of bioinformatics is because we want to develop our own system or pipeline for analysis of our bioinformatics data. Now, as we discussed last time, the word pipeline comes from the pipe command in Unix, which a lot of you have used already. It was part of the homework. So what it literally means is the input of one goes to the other. That's why it's a pipeline. So today, we will, let's go over the first slide for today. Everyone has the Unix accounts except for a few people, so we will go over that and try to cover that as part of the practical. If you don't have it, no need to panic. We will still we will still try to help you out as much as we can. The issues part we'll go over. Now, for the feedback from last lecture, I did receive most of your comments, so we will stick to what I have learned from them, including a break after an hour. So I understand we all have needs. Get it? So all right. So let's proceed with the lecture then, and then we will cover the Unix or whatever issues you have in the practical section. Alright, so let's talk a little bit about clusters, not the ones that are yummy, the other ones where we do our work with. So, IRS1 cluster, I know we've talked a little bit about it last time. So, you have been trying to apply for an account, some of you have been successful. Let's start with a little bit of the basics, some of which we covered, but just to revise what we covered. So, a job is a command that is submitted to the cluster for execution, and a queue, so it's a software line. Believe it or not, in a lot of countries outside of the United States, a queue literally means a line. So if you're saying I'm in the queue, you wouldn't say actually I was waiting in line, you would say I was in the queue, right? So, oh, you jumped the queue. So that's where the word comes from. A line of jobs is called a queue. And so a scheduler or scheduler, depending on where you're from, is a software that manages which jobs run on the queue and how much resources are allocated to that job. Now, scheduler is a software, so it's not a person trying to give you priority or something like that, but the priorities are inbuilt. So it's very much like a person who tries to, manage your jobs. So think of it as someone, depending on what your arrangement with the HPC cluster is, they'll give your jobs priority. So, for example, our lab has purchased some cluster nodes specifically, or the Rama lab has some clusters that are specifically for their labs, so they get priority on those nodes. So they have, say, 10 nodes or 10 computers. So anyway, so that kind of priorities can be managed by a scheduler. So this is where you were to read a little bit about the IRS1 cluster. We will learn a lot of this stuff on this as part of this course. You can just Google IRS1 cluster and then you will be able to find out what are the rules and some of the things that were required. So one of the main things that I wanted to go over, I'll cover the summary, but I would highly recommend that as part of your homework you go over that, the previous link. So this one, alright? Again, the slides are available already online, so you can download from there. Or feel free to Google IRS1 cluster. So login nodes are not for running any of the programs. Now I know I mentioned this in the passing, yes, last time. But if you do, you will get something like the howler that we got in Harry Potter. You get something like that from Jonathan Jackson, and not the real kind, but it's pretty nasty. So we use the compute nodes for submitting jobs that are data intensive, and login nodes are for running scripts and test programs on small data sets. So by small, so there is a limit. So you can run up to 100 MB on data sets. So don't run like an alignment, for example, on a 2 GB or a 3 GB data file, right, on your login node. Now, if you're confused at this point what a login node is or what a cluster node is, ask, and I can clarify that. Or shall I clarify that further about what the login does?
Yeah.
Okay.
Yeah.
All right, thank you. So when you log into your IRS1 account, you essentially log into a computer. Think of it as a gateway computer where all users can log into. Now that gateway is your login node, and it's also the gateway for all the other users. So which is the reason why it is not programmed to handle high memory or high intensity jobs. So behind that login node, there is a huge number of clusters. So that's the cluster where it resides, right? It's also a matter of privacy. It's a way of kind of separating your cluster from the world. So you create a gateway server or a gateway computer, and those are your login node. And then behind that is your computing node. There's a specific way of reaching the compute nodes from the login nodes. And again, we will cover that in just a moment. So the login nodes, when you log into, are ears 1 and 2, ears 1 and 3, literally ears 1, node 2, node 3. So they have multiple login nodes because there's a huge volume of users. And you're randomly assigned to a node for the purpose of running jobs or accessing your files. They're very transparent. No matter which node you log into, you will still have access to your data and your files. You wouldn't even know. If you didn't notice N1, N2, N3, you would not have any way of knowing that you're in a different node. Okay. Now, behind the login nodes are the queues, are the queues which are your gateway to your cluster. So, login node, scheduler, where you submit your job to, which then puts it in this line of jobs that are waiting to run on this big cluster behind. Does that make a little more sense now? Okay. So I like to think of it as an airport. When you just get into the airport, you know, you wait in, it's like the check-in process. The first node where you check into is your check-in terminal. You know, you buy your boarding pass, you get your boarding pass, you get your ticket, and then you stand in line where, you know, there's a huge line. Those are your jobs. And then the TSA agents or your schedulers, they can come and tell you, no, you move to the back, or you go to the priority line. Oh, you're with children, you go to this line. So that's your scheduler, right? So it's very much like an airport. And once you go through that security, that's the process where your job is running. And then once you're past that, you go through the scanner and everything, that's when you're done, your job is finished, and you come out on the other side as a finished job. Okay, does that analogy? It's like that's exactly what this is happening. So the cluster would be the scanner. When you're going through that scanner, you're actually executing the job on that cluster. Okay. All right. So these are the different queues. Now queues, like lines, or I said at the airport, there's one line for seniors, there's one line for people with kids, there's one line for people who require a lot of time, there's a business express line where everybody knows what they want to do. So just like that, you have different queues for running your jobs. You have the very short queue, where the maximum runtime is 15 minutes. You're an experienced traveler. You're able to get through the line really fast. You know your job is not going to take more than 1 GB of RAM. 1 GB of RAM means 1 GB of running memory. So all the processes that will run when you run your program, when you type your Unix command, you have some idea that they're not going to take 1 GB. 1 GB is a lot of memory, just so we know. Before we had these laptops with 16, 32 gigs RAM, 1 GB was considered like, whoa, that's a lot of RAM. So short is the second queue, which has 2 GB of RAM as the maximum memory, 1 hour, not more than 4 GB. So 2 GB is the default, and then not more than 4 GB. And you can get 1 computer per core job. So you do not need to have parallel jobs here. Or no more than 1 node. So you're not going to run jobs in parallel, you just want to run 1 job requiring 1 computer, right? Not that most of the times you will be using a lot of parallel computing, but this is what the requirements are. Then the medium queue is 2 GB with a maximum of 24 hours time. So here it's 1 hour time, here's 24 hours time, and 8 GB is your maximum memory that you can run. So now this queue is default for most processes and is pretty much sufficient. So with this much RAM and 4 processors available, if you need them, you would be fine. So now the reason it's important these names are in capital is because when you submit your job to the cluster, you have to actually specify that this is and it's part of your command. So as part of your command, you will specify that whether this is I want the short queue, I want the medium queue, or I want the normal queue, or I want the other queues, which we will talk about. Anyway, so they just differ in the number of times. So like this is also 8 GB normal queue, but with 3 days maximum runtime. Now this comes with a little practice. So once you submit your job to a cluster, you may get some sort of email, nasty email, like a howler saying once in a while, but you pick up really fast that if I anticipate that it's going to take me 2 hours, I will just submit to 24 hours, you know, cluster. Yes?
So do you suggest, if you're not really sure, to always do the shorter one and then,
no,
or
I
to
suggest,
do the longer?
Yeah, just be on the safe side. Just put it for, unless you're really short in time, like, you know, you're like, oh, I need this in 15 minutes. So because if you submit a very short job in the very short queue, then pretty much it's guaranteed it'll run right away, which means that even if there are 20 jobs running, they'll give you priority because you're saying it's a very short job.
And if you don't have enough memory, do you get an error message, or just
yes,
not complete?
No, you'll get an error message, and it'll basically, there'll be a core dump. It'll come as part of your email or whatever output you specify your error message to come to. We'll go over that, but it'll come and say that, you know, your job failed because of term swap, TERM swap, SWAP. That means that there was a terminal swap, which means that it couldn't get enough memory from the system and swap it in place. So there was too much swapping. That's an operating systems term. It's more of an advanced concept. But suffice it to say, what the computer starts doing when it starts running out of RAM is it starts to struggle and create these virtual memory boxes in your hard drive. So that's why sometimes your laptop will become really slow when you have a lot of programs running. So because your RAM tends to fill up, so then what the terminal, what your computer does is the processor creates a box of virtual memory on your hard drive. Now hard drive is a lot slower than the RAM, and that's why it starts to bring files back and forth between your hard drive and your thing, because that's where it's storing all the information now in order to run. And so at one point the cluster will be like, okay, I give up, and it'll just crash. Okay? Okay, so then just, if you need more than what we just discussed, there is more options. So you have long, you have very long, and with very long you get 2 GB with a maximum of 4 weeks of time, and 2 to 4 GB maximum with 4 nodes, and same thing, 2 to 8 GB with the long queue, maximum 1 week. So, you know, and then there's a big multi-threaded, so you can get up to 40 GB of RAM on the cluster. Or with big multi-threading, you can get 40 GB, and you can, you know, you can also get more than that. If you email them and you set the memory limit with them in advance, you can get more than 40 GB. I do not anticipate you needing it, but that's a possibility. So again, you don't need to remember all of this, but if you are ever in need, know that these options are available to you. Okay?
Are there like costs associated with each option?
If you're part of the cancer center, then no. If you're not a part of the cancer center, then there is some cost, but I believe the Broad has their own servers exactly similar to this, and so if you talk to the system administrator just like we have here, they will be happy to give you some time on their clusters. Or you can tie up with the partner's cluster for a cost, yes. Okay. So this is what it looks like, you know, go through, this is that page that I mentioned. These are the FAQs, yes?
So there's no way the computer could estimate the GB that you're going to be using based on some...
No.
You just have to do it based on instinct. Or
well, a good rule is like if your file is 2 GB, don't run it in the short queue. Right? I mean, if your file size is going to be like that, because certain programs, I would just double or triple my file size most of the time. So that's my basic thing is, like if you know your data file itself is about 6 GB, and you're aligning, so aligners most of the times will load the entire genome. So also matters is what you're doing with what kind of genome you're aligning to. If you're just aligning to a Arabidopsis thaliana, which is like, you know, a couple of MBs, or you're aligning to the human genome, which is GBs. So an aligner will load the, as we'll go over in a bit, you know, in the alignment section, that aligner will load the entire index of the genome in the memory. So that memory would be a huge memory, especially aligners that are fast. So star aligner is a newer aligner, it's really fast. It aligns in about order of magnitude faster than top hat. But the disadvantage is that if you're running it on your local machine, you won't be able to run star because it loads the genome, entire genome index onto your memory and then aligns the sequences. So that's why if you're aligning with that kind of aligner, make sure you allocate more than enough. So like when I do my alignments, I use the big multi-cube. That's what I do. But that's also because my files, you know, I get like huge data sets and I try to run them in parallel so as to save time. So I'm done with 100 jobs in one day, most of the times. Okay, so Now, we talked about the cluster or the server. So we talked about the softwares that are available. Some of the Unix commands that you studied last time, those are also part of the software, but they come as default with all servers. So Unix is like a default because that's what the server is based on, so they come as default. But for bioinformatics analysis or for any tailor-made analysis, there are certain software that will not come as default. So you will have to either install them. So I had one of you try to install a software. It's part of the homework assignment, that muscle software. And when you type the command on your own computer, sudo apt-get install or sudo install this software, that's a way to install your software from the command line. So it's just like downloading a software and then installing. Now in Unix there are certain repositories online. So think of it as a free database available to you, and your operating system knows the location of that database. So when you type sudo apt-get install, it means that you're saying as a super user do, that's sudo. Apt-get is the program that is saying get this software from the repository, and install is the command that you want to do with that software, and then the program name. So it'll search the existing repository for the program name and install it. Now, this is in Unix. Most of the times in you will not have to deal with this problem because the people in the cluster A don't want you to install software, because as users who have not gotten that much experience, they don't want you to install. In fact, if you try, sometimes again you can fail and then they will give you a nasty email or something. But the other option is this module load command. So they have already installed pretty much most of the bioinformatics software that you need. So most of your bioinformatics software is already there, including your aligners, top hat aligners, tar aligners, quality control programs. So all of that is there. The way to access those is to use this command module load and the name of the software. And again, in the practical, we will be doing one of that. And in order to use the module command, which is itself a software, right? So remember in Unix when we talked about everything is itself a program that is written by someone else, even the whole operating system. So even a module command which helps you load other softwares is a software itself, right? So what would be the biology equivalent of that? Like, what are there are some genes like that are basically there to help other genes, right? Or proteins that help other proteins to do their job. So this is the same thing. So module, think of it like that. The module is that kind of command. So when you do this line and add it to your, when you're on IRIS1 in your account, you only have to do this one time. So the first time you get your IRIS1 account, you log into that server, and after logging in, you add this in your .bashrc. So in the last lecture I showed you these dot files, the .bash profile, .bashrc. They are stored in your home directory, and these are the commands that are executed before everything is initialized. Think of it like a booting, like when your computer boots up, these, or when you log in, these commands are executed. So anything that you add over here will be available to you for the duration that you're logged in, right? And when you log out, it doesn't matter, but when you re-log in, again, these commands are executed. So what you're doing in this command is saying module use apps module files /lab, and then you're appending it to your bashrc, right? .bashrc. So I know it's a little bit of a difficult thing to grasp right now, but basically echo command, which was part of this, is echo means just whatever I type, just regurgitate it and print that out, right? That was echo. Okay. And in codes you're saying echo this command, whatever is the output of this, append it to bashrc, which is basically it's going to type module use this and append it to your bashrc. Okay? So we'll do this. I'll show you what it means in a second. Again, in the practical section, we will go over this. But if you just want to get the gist of it, it is that we're trying to add this line to your bashrc so that it's executed every time you log in.
And you have to define your variable ahead of time?
The $home variable is already defined.
Oh, is it?
The $home always points to your home directory,
Okay.
which is why $home is like, it'll be
Okay.
/phs home in IRIS1 /lnieman or whatever your user ID is.
Okay.
Okay. Now, after that, once you have done this, now you have access to all the software on IRIS1. So you can just type module load, and if you press plus tab, like the tab key, then you have access to a list of software available. We'll do this in a second. Would you like me to do this now and show you? Would that be more beneficial?
Yeah.
Okay, let's do it right away then. So, I agree. Okay, so let's go.
I think it's just that. It's like
I just tab.
Do you
Okay.
have to exit and log back on for that to load, or is it already loaded?
For you, if you haven't logged in the first time, this command, if you haven't typed this command earlier, it won't be available.
So if we're currently logged into IRIS1, we should log out to do this, or?
You don't have to log out, no.
Okay.
So here is the command. So basically,
So there's a space between module and use?
yes. And there's quotes, don't forget the quotes. Right? So in my case, let's see.
Oh, there's quotes between module and use.
Oh wait. Okay, so right now, where am I? I am in my own home computer, right? So this is, I still have to log in. So the login, the way we log in is SSH at, okay. Now last time I created an alias for this, but I'm using the full thing this time just to show you this is how you do it. Okay. Now, notice as soon as I logged in, what do I see? I get all these, these messages which I recommend going through. But as I was talking to you earlier, we log in the first. So I have been assigned IRIS1 node 2 today, right? So again, this doesn't matter much, but it's just I'm saying that this is what I've been assigned to. Sometimes it's node 3, sometimes it's node 1. Okay, so next step. Now let's see what is there in my bash RC, right? So see, my bash RC already contains this line, right? That's because the first time I logged in, I ran this command like this, exactly like this. So, but in order to show you what I'm going to do is I'm going to go up and I'm going to delete that line, okay? And now I'm going to save it and leave. Now, this is the line that I had said, right? So I typed it out, executed it, right? Now let's go back to...
Can you remember what Vim is? It's just an editor.
Oh yeah, so Vim is just an editor that I'm using. Feel free to, well, right now okay. So VIM is a primitive editor, right? It's something that I use. Again, it's from the times when I learned and there was no GUI editors. So this is a simple way to edit stuff in your, within your thing. I would not recommend you start using it now because then we'll go into a tangent and go into a whole different world where I have to teach you Vim and how to go through the commands that are there. So don't do this right now. In case you're doing and you want to exit, just do escape :Q exclamation point. Okay? So
if you're in Vim.
if you're in Vim. I hope you're not in Vim right now.
I typed that vim-rc, yeah.
Okay, you typed that.
I did.
That part is fine. So if you're logged into IRIS1 and you type this, that means that it was added. So just as you can see, notice earlier, this line was here after alias, right? And now it has been appended to the end of my file, because that's what the instructions were. Does that make sense? Right?
No.
Yeah.
Okay. All right, let's go over it one more time. So, this command right here. What did I do? I typed echo quotes this greater than size dot home bash rc. Right.
Why the quotes? Is that to
Yes,
escape
yes.
the
So
spaces?
the basis for this. So okay, so I'm going to just exit this. The way to exit is :Q, exclamation. After press. So first we hold on. So first we press escape key :Q, exclamation. So this is not your standard editor. So you don't need to use Vim. You can use Text Wrangler right now, and that takes care of all of this. It's a GUI editor. But all right. So let's first go over echo. What the echo command does. Here, if I type echo Vishal, it types Vishal. Okay, so that's the basic reason why echo is just like a way to, echo back what you just wrote.
Is it similar to print?
Yeah, it's similar to print, but it prints on the command line. So echo, this is bio, whatever. So it'll just print that, right? So now, as we see, this is the output of this, whatever is in quotes. Now if we go back, what is the output if I remove all this? The output of this is just module load this, and it prints that, right? So what we were trying to do is append that to this. Now, another thing we can do is, if you want to know what home points to, we can do echo $home. Now, $home translates to this, which is the home directory that I have here. Okay, so we've broken down the command now into multiple parts. Okay, so $home here, you could either just write /phs home /vt094 and then do this, or you could write $home and because it's going to translate to that. Okay, and what it's saying is echo this, whatever is in the brackets, and output it and append it. So remember the two greater than signs are append. Append that to this .bashrc.
The dot specifically.
Dot
means that
is
it's a
for
script.
system files.
It's a system file.
So system files are executed when you log in. Every time you log in, anything that is in .bashrc or .bash profile will be executed. So if you want to see what system files you have, you could just, in your home directory, you could just do ls -a, or, so see, I have .fontfig.config.git, .java, .less, whatever, .elicit bash, like these are directories, but you also have these files, .viminfo, and if I did this, and :more, so now you can see all my files show up earlier. So you see this .bash history contains the history of all the commands. So when you type the greater than sign, if it's not in .bash history, that file, that command will not show up. So it is a history of all my commands. .bash logout is whatever info is stored at logout. Profile is again a set of commands that I have for my profile. Like I can choose to put these. This is where I store my personal command. Bash RC, again, my system commands that I want the system to execute, I store here. All of these are executed when you log in.
And they're all created by default.
They are there
Bash.
in every Unix system by default. These are there.
And we want to be a bash and not sh or whatever, right?
Because
So
we're
there's
using the
different-
bash
there's different
shell. So again, uncomplicated Unix. Simple thing is like there are different kinds of shells. Remember we talked about the shell last time. So this is the bash shell that we're using. It's just a way to access the inner Unix. So that's it, and these are your files. And you can create your own files like this as well. If you want to add some commands to bash profile, feel free to do it. Those will be executed every time you log in. Feel
and
free.
if you wanted to see what was in bash,
Yeah,
you could
so
use Vim or text
you could
editor,
use text edit.
Wrangler.
Yeah, you could use text wrangler, or you could just go, if you want to just see, you could just go less or cat dot bash profile. Okay? So this is what my bash profile contains. Starting from here. It says this is a hash is a comment, so these are all comments, but then it says if bash rc has it, then execute bash rc and then go and execute, otherwise end of if, after bash rc execute this. So it's saying, first thing it's saying is append this to the path. So your path is where your files are going to be searched for when your computer starts executing your command that you enter. So remember, bin directory is where we store our commands. So the first thing you want in your command to be like, at least give me access to all the commands that are there in Unix, without which I will not be able to do anything. So bin is first thing is added to your this. Then these are the other things. So Apache server is started. You export the path. So again, these are all commands from Unix. I don't want to go into this, but there are some aliases. You think of it as, you know, like a user preferences, whatever you want to add.
Okay.
You know, in our case, we want to make sure that we have access to okay, so we're out. But yeah, we want to make sure we have access to this. So anyways, we'll do this. Don't get carried away right now. Yes?
Quick conceptual question.
Yes.
I'm lost on something.
Sure.
What's, like, conceptually, what's the difference between bin and bash? Like, the way I'm seeing it is, like, sorry if this is like a ridiculous question, but bin, I think of it as a directory where you're keeping all your commands, and bash is, how is that related? What's their relationship?
Bash RC, the file.
Yeah.
So, yeah, so one thing could be you can load put your module file, okay, so module is the command.
Yeah.
You could actually store it in bin, and then it'll still be fine. You will have access to module as a command. But you want to make sure that this command is saying that use this particular repository for finding the softwares that I want to use. So what this module command on itself is going to be stored in bin or whatever, you know, which is there in your path already. But when you say module use this repository, you could have created other repositories as well. So, okay, back off. So back up a step. A repository is a place where you store all your softwares, right, which you want to run. Now for EARSFun cluster, they have created a repository of all the frequently used bioinformatics software and kept it here in this directory, apps, module files, lab. Okay? Are you with me?
Yeah.
Okay. So if you wanted to use your own software, you can create your own repositories as well. I do not recommend you start doing it right now, but when you're an advanced user, feel free to do that. In which case you can write another command saying that module use, don't use this, use my repository because I have my versions and I have my software. Till then, we'll use this repository, but module, while module is the command which is stored in /bin directory, I still want to specify when I run module what repository I want to use to load the other software.
Okay.
And that is what we do here.
I see. Okay. Thank you.
Okay, is that clear? Everybody? Are we still on the same page? Okay. Alright, so we're going a little behind. So let's talk a little bit about pipelines. And what is a pipeline? So by now you know we talked a little bit about this. I'm going to skip this. So, okay, so now we're going to start learning about, straight away dive into this analysis part. We'll take it one step at a time. And so, you know, don't panic. We're not going to do all of this today. This is going to be for the next 3 lectures. We're going to try and cover this entire thing. But today, we will give you a taste of what it is like to create something. And the RNA-Seq pipeline, as a case study, uses the following things. So we usually start with FastQC, which is a program that analyzes your quality scores. We'll go over it in a second. We'll understand what quality scores are. And then we use the alignment software to align our, after we satisfy the quality, we take our files and we align it, and we start, and then we count the reads that have aligned to specific regions of interest, so genes or exons, whatever you're interested. And the output files are, you know, you have FastQC files, you have aligned BAM files, and then you have a count table. And then further downstream analysis of the count table happens in R, or if you want, you can do it in Excel or whatever software you prefer, or MATLAB. Okay, so let's start. So FastQC, this is what we will try. Hopefully we'll get to the end of it in the next 20 minutes and cover this. We will also cover these softwares later on. Okay, so let's start at the very beginning. It's a very good place to start. So let's start with the file formats. Now, when you're going into bioinformatics, this is an unavoidable sort of evil. Because bioinformatics was not an organized sort of discipline that came about, like everybody sat down and be like, all right, let's create bioinformatics. It wasn't like that. It was very much like everybody over there started creating something, and then people at WashU were like, oh, wait, the UC Berkeley has done this. We gotta get in line and start reading. So everybody created their own formats of files. And then guess what happened? Like there are tons of file formats, and this is the reason why we have this class, is because otherwise you'll have one format, one way to do it, and I wouldn't be needed. So anyways, so there are different formats for the sequence files. So the sequence that comes off your sequencer. Then there are file formats for your alignment. Once you're done with the alignment, all of these are related. And then the most important is the way your feature formats are for your reference genome. So your reference genome has to be in a particular format in order for for you to do the counting of your reads. Okay. So, quick overview. This is how a FASTA file looks like. Okay. Now, without going into further detail, this is just you can take a look at it. There's a sequence ID, and there is a sequence. The sequence ID could also look like this. It can have characters like pipe-separated characters, which tell you something else about what the sequencing was, what the reference was, blah blah blah, group was. But not almost, always, you will have one line with the greater than sign saying this characters. This line is the ID. It is ignored as far as the sequence is concerned. You can put anything in here, okay? But the sequence, the actual sequence of your aligned reads is the next line, right? And onwards, all the lines, until you hit another greater than sign. Everything in that is your sequence, okay?
So that first line is just informational, it's like a header.
So it could, so this is like a genome sequence.
But it just tells you information on
Oh yeah,
where,
it's
when
just
it
exactly. That's exactly what it is. It's just information for the purpose of, because when you're a programmer, you'll go to this line and you'll be like, oh, okay, this is what it is. Otherwise, I have no way of knowing. Okay, now the FastQ format, on the other hand, is mostly used for reads. And that format has a different format, as you can see. It starts with an @ sign or a unique sequence ID. You have the sequence, then you have a plus sign, and then followed, which tells you that the next statement is going to be the quality. So these are different variants. The Sanger will have a different format, but they always have 4 lines. So instead of having 2 lines specifying what the sequence information is, they have 4. Because here, you also want to have the quality call for each base. So this is the ASCII symbol for the quality call for this base. The plus sign is just a break. It tells you that the next line is a quality. Okay? So 2 lines identifying the sequence and the identifier, and the next 2 lines identifying the quality. Okay? Is that clear?
So what does that quality stuff mean?
Right. Good question. Very good. So we can skip over. Anyway, so just quick answer to that. I'll get into a second, but these files can be very large, sometimes hundreds of GB at a time. This is why when we use the queues, we have to specify the memory limit depending on how you sequenced, what was your depth. You can have a lot of data in this file.
And what comes off the Illumina sequencer? Is it FASTA or FASTA or both
FastQ.
of these?
This FastQ files. This is what you get from your Illumina sequencer. Exactly like this, with some variation, depending on the quality score. Okay, so we'll get to the quality score in just a second. Before we go there, I just wanted to cover a little bit about the reference genome that we have. So if you're a human or whatever, or exons or introns, or almost always you'll have something called as a tab delimited file to store genomic features, intervals of genes, and gene structures. So these are essentially like this, columns where you have chromosome ID, your source for that, your gene feature, so it could be a gene, mRNA, UTR, exon, start and end of that on the actual genome, followed by some user-defined score, a strand of that, the phase, and then the attributes. Again, these attributes can be user-defined. You can put some stuff here. This is the GFF3 format for
what's the
reference.
Phase? What's the phase? What's phase?
I'm honestly not sure about that,
Okay.
but we could look it up.
It's fine.
It's usually something that we don't usually care about. I mean, as a bioinformatician, the only thing you care about is from here to here.
Sure.
Most of the time.
I just didn't know what.
Yeah, no, I haven't used it. I can look it up, honestly. I'm not quite
It's fine.
sure.
It's fine.
So you have the chromosome ID in the GTF. So GFF and GTF are essentially the 2 file formats used to represent the same thing, which is the genome or the chromosome or the genomic features, but they have slightly different formats. So we'll see when we put them together. Like here, as you can notice, there are slightly different especially in this field. So here you have a gene ID, a transcript ID, and it's followed by these colons. Here you don't have that information, although you could add that. So they're interchangeable, and most softwares accept both these formats. But most of the times when you go to UCSC Genome Browser to download, you download in this format, and this is what we use as bioinformaticians most prominently, is the GTF format. Okay, now coming to, oh man. Okay, so before we move on, I'd like to answer. There was a whole section where we go over the quality scores, but we'll just get there, or we can start with this now. But I wanted to do this joke before. So quality matters, right? So Nicole, this was for you. So no quality scores for you. If you don't, so these are quality scores for that we just talked about earlier. And the sequencer outputs base calls at each positions of a read. So basically when you're aligning a read, you know, or when your sequencer is reading your read, not aligning, when you're reading, it's making a call, right? Now that call is a probabilistic call. Like with most things, it's not really certain whether this thing is 100% an A or a T. It could be something with a high level of confidence that says, okay, I'm 90% sure that this is an A, right? That's how the sequence calls are made. So that quality score is what essentially you see in those characters. Now, there is an encoding to that, so that probability is encoded. So if this is the probability, then what we do is we take a log 10 of that probability, because probability is usually going to be between less than 1. So we take a log 10, and then we multiply it with minus 10, okay, to create a Q Sanger. So for example, and this is a call of which is incorrect, so there's probability that this is incorrect. Now, there's a logic behind this. The reason is that we wanted something that would be, that would tell you, like if there is a point, if your probability of making the base call is 0.05 for it being incorrect, not correct, incorrect. So then there's a 5% chance or 1 in 20 chance that it is incorrect. Or if it's 0.01, then it's a 1% chance, or 1 in 100 that it is incorrect. And 0.01, then same, 1 in 1,000 chance is incorrect. Using the equation though, it translates to 0.05 is Q of 13, 0.01 is Q of 20, and then 0.001 is Q of 30. So what it means is, any direct inference from this?
The higher the better.
Number, the better the quality.
There you go, perfect. So that was exactly why they did. Now do not ask me why they didn't go the other way around, where they said like, okay, why couldn't they have the probability of being correct? And then it would automatically it would be higher. You don't have to multiply by minus 10 or something like that. This is just the way it is because they came up with this. The person was like, we'll do the incorrect one first. Yes.
How do they actually generate the quality score? Like, what are they going off of to make that number?
Right. So that is built into the calling software that Illumina uses, and some of it is proprietary. We don't exactly know how they do it, but basically the Illumina, when it's reading the base, it's making the base call, right? Or Sanger sequencer, whenever the Sanger sequencing came out, they were basically making the base call, right? So it's an intensity of light. So Linda would probably know more about this when it comes to light intensities, but it's always a probability, right? So you have some value that this call is this much percentage based on the light that it emitted. So if the A frequency is this much, then you'll be like, oh, the A frequency is this much, but we are also getting a slight T frequency. So because that's how the read calls are made. So you're like, it's 90% A, we'll just call it an A. But you have to still make the call and say like, okay, there's a 10% chance that we are incorrect. And that's how they make that call.
So they've got some magic mix that they use to make this prediction?
Pretty much. I'm not quite sure exactly how it is done, but that's what we understand it as.
So when you get a whole string of whatever sequence, let's say it's 100 base pair long, do they calculate the quality score for the whole?
For each base pair.
For each base pair, so not for the whole?
Not for the whole thing. That's up to us. So we do that later on.
Okay.
Okay. So,
are the quality score comparable among all these sequencers?
So we have a way of basically, if you have one kind of sequencer, right? Yeah. So you will be using that formula for Sanger sequencing. So that Q you can calculate the same. The Illumina sequencer will have a different quality metric, but they are comparable in that sense that they will use the same -2, and you can also interchange between the two. So you could be like, well, I want the equivalent in the Sanger quality score for this thing because I have one data on this sequencer and another data on this sequencer. That said, would I compare them? I would not be comfortable comparing them because in the sense that there are a lot of intrinsic variables that I don't want to account, that we are not accounting for, even if you transfer the quality from one to the other.
I see.
So it's not strictly standardized.
Exactly.
I see.
So what is the acceptable and not acceptable scores?
So we'll go over that in a second. So if you're already way ahead of the class, or last time we taught this after we had done R, so we moved this section before learning R. Just because I thought that it's more in sync with what we're doing in Unix. But earlier we did R, so there is a function that I wrote in R that you can, it's actually publicly available, it's not I wrote it. But it's a very simple function, it's just basically the same formula. You can write it as a function and then you can calculate these and you can plot them and then it gives you a very nice plot like that. The other way around you can do as well, so if you wanted to convert your quality back into that. Anyway, so basic thing to remember is Q30 is 1 in 1,000 chance if it's correct, Q20 1 in 100, that kind of thing. So we will go into quality encoding, but alright, let's. Okay, so this is where we go and transfer your file formats back into what it is. So Q squared of 20, 30 takes 2 digits. So like 20 is 2 digits, 30 is 2 digits. Bioinformaticians and computer scientists prefer that it only take 1 digit. So in order to save space. So therefore, they figured if we use characters instead of numbers, it would make life even harder for people. So why don't we do that? That's how you get the characters from the quality.
But there's less memory though.
There's less memory though, exactly. So basically they add a number to it and then they take the corresponding ASCII code. I think the number is 64, we'll find out in a second. So add 33. So you add 33 to that, and therefore you get your character. So let's say the probability the base is wrong is 0.01, which comes to 30. You add 33, and the encoding character becomes this question mark. So every character on your keyboard has a corresponding number.
Can you go back to the key?
Which key?
The previous one?
Yeah.
I like that. So geeky, I love it.
Where's 1?
Huh?
Where's 1? It starts at 33.
Right, so that's basically, that would be a very low quality.
Oh, okay, you're not going to go below that. So,
right. Well, you probably won't get a 1 in because that would be horrible.
Ignore it probably.
Yeah.
Okay.
But anyway, so we didn't have it 1 over here, but that's how we convert from a probability to a character. Okay? And then there's a function for that. You have a UTF to integer, and when we learn R, you can feel free to go back to these slides and write these function out and try it out on your own. Fun stuff. Okay, so we're almost out of time. I'm going to quickly rush to this. FastQC is a free piece of software that's there. It was written by Bob Braham's bioinformatics group. It's available here. It's also available in our EARSON cluster, so you don't have to download it if you want to. And for the purpose of your own thing, if you want to run something on your computer, feel free to download it. So when you run FastQ, it's very simple. You write FastQC, the program name, and the name of your FastQ file, and you press enter. That's how simple it is. The only thing you have to type before that in the EARSON cluster is module load FastQC. Okay, so we'll go over that. It's part of the practical, but that's what it is, and then it generates this beautiful output graph. Now notice it goes all the way through here. There's a green section, there's a brown section, and then there's a red section or a pink section. You want to be in the green section. This also gives you a bit of an idea about the standard deviation. Now this is where you get the quality for the entire file. So what we've done is we've created box plots for each base. Alright? So as you can see, very much that initially till, then this is an older data set, so you only have 40 base pair sequences. Now you have much bigger, but the graph looks very similar still. The quality drops as you go towards the end.
So why does the quality drop as you go towards?
But that's just in the machine, so
the
yeah.
machine just degrades in its
that's
ability
the chemistry
to
of
be
it.
That's the
accurate?
chemistry, exactly.
The chemistry.
Yeah.
Oh, okay.
Yeah.
See, I have no idea.
Yeah. So you will notice that with pretty much all samples, so it'll go down. Now the question is, to what degree does it go down and where does it start? So if it starts right here versus it starting here, that makes a huge difference. And then how much does it drop? Like, do you have a huge thing like this, or does it suddenly just like all of it is centered here? this is your range, and all of it is bad. So based on that, you have to make a judgment as a bioinformatician whether you want to keep this sample or not. Now, it's a tough judgment, but you have to make it as a bioinformatician, or you can go over with your bioinformatician and be like, oh, okay, this is what it looks like. All right, what do you say we do? Shall we? One option is you can chop off your sequence reads from 20 base pairs and be like, or 40 base pairs from where it starts, and then we're like, all right, we'll just think of these as 40 base pair segments and align those. We really don't want to have the low quality ones because I really just want to have the best calls. But that's entirely up to you. So, yeah, so, you know, it plots a box plot, shows the distribution of quality, positions all across. So here is something as an example where I would just be like, oh yeah, let's just throw this out. I mean, we don't know what this is happening here.
Because of that fourth point?
Yeah, I mean, it just dips at that point, fourth position, so like, I don't know what's happening, and it goes all the way to here, and then these are the only few bases where it gets better, and then it gets, again, drops dramatically. Less obvious problems, I mean, this looks okay, but again, still it kind of drops pretty significantly from that point, and then really bad problems in this case, alright?
So what do people do to try to make that better, to make the quality, you know, the good area longer?
It's got, I mean, it all depends on your library prep, it depends on many different things, and the sequencing and, you know, kit that you got sometimes is variability there, so there's a lot of issues that could create all this.
But the important thing to know is that, you know, as a biologist or whoever is working on your data set, you want to make sure that you run this and see for yourself. You know, it'll give you an idea of how your sequencing went. Right? So you could do a couple of samples, sequence a few samples, less samples, and then go over them and be like, all right, this turned out great, so now I can do another run. Save yourself some money, time, you know, all that.
But is your goal always to keep it in the green?
Mostly, I mean, you know, towards the end, if it's like this, I'd be like, yeah, this is okay. I'm not gonna throw out this sample.
You would align all 40?
No, I'd align all 40. Yeah.
So green is the 0.001, right?
Yeah.
And then the brown is the 20 to 28, so that's like 0.01, I think, right? Yeah. So you want to keep all the points.
Yeah. So as long as it's over, you know, most of them are fine, like, you know. Brown. And then the other thing is, so this can also be corrected in the alignment stage. When you basically go over your aligner, you can specify parameters saying, okay, I can allow up to these many mismatches. I'm okay with, or I will just throw away reads that have more than 2 mismatches. So if like like this sample would just get thrown out. Most of the reads from this sample will get thrown out, even if I align it, because in the alignment part I have a check. 1 second, let me finish and then we'll go over. Okay, so that is, well, the last slide for today. So we will stop here and go over getting to the server, running some FastQC. I did skip 2 slides. If I have 1 second, I'd like to go over that. Those were basically these two, the SAM format and the BAM format. So this is the alignment format. I think we will go over this, it'll make more sense once we start to go over the alignment actually. So, but just to give you a quick preview, it stores the alignment information. So just like the other formats where there's some information stored regarding your file, the alignment, once this has happened, it stores that alignment information, like where it aligned to, how was the quality for that alignment itself, what was the, you know, is there multiple match, is this read multiple mapping, or is it just single mapping read? So all of that information is stored in this format. Now, these files can be very large because it stores more information on top of your FastQ files, because now it has the alignment information as well along with the other information. So therefore we compress them and convert them into this BAM format, or binary alignment format. And that's the BAM format. It's not readable by the human eye. So whenever you have to read it, you have to convert from BAM to SAM, but it's 1/5 the size of a SAM. It's still very large. But we can just basically, that's where Unix comes into play, where you can just be like, you know, you have a software that goes from SAM to BAM, and you can just pipe that output and grep and do all that magic and kind of get your, you know, information the way you want. Okay, so that's pretty much it for today, and we're gonna take a break. We'll start going over all your issues, and then before we get there, are there any questions? Okay, go ahead.
Are there like best practices that people follow, or like some codified document of like if you want to have like high quality analysis is what you do?
So I don't think there's a pre-document. There might be some with the Illumina manual, or yeah, I think a lot of it is practice. As you know. There are some, you know. Yeah, you would know better.
Yeah, I know a little bit. So there are some things that people do, like best practices type of thing. Like you allow for 1 or 2. When you're doing the alignment, you allow for 1 or 2 mismatches. Some people are very strict, they allow for none. Some people, you know, it depends on your quality of data, right? So if everything is in the green, you can be much more strict when it comes to your alignment. And also depends on how long it is, right? I mean, that also makes a big difference. So some people allow for 1 or 2 mismatches. That's normal. 3, I would say, is very loose. That's when you get into trouble. But generally, that's general practice.
Right, so that's the best practice that you follow in the bioinformatics analysis. Before that, like how can you get better data by just your, by virtue of doing the prep? That part, I'm not very familiar, but I'm sure Illumina or whatever sequencer you're using will publish some guidelines. I would go talk to, you know, the technicians. They're the ones who actually do that alignment, so they might have some hacks that, you know, we're not aware of. Okay, all right, so we'll take a 5-minute break, and we'll convene exactly in 5 minutes. Perfect.
So it doesn't let me, it says permission denied for me. I don't know why.
Yeah, permission denied.